Hello. Yesterday we conducted the first field experiment to ask questions about the experiment design for collecting samples for fungal metabarcoding in the peatland, in the bog. And we asked several questions about the extraction approach, about the weight of the specimen, whether the specimen should be single or composite, special arrangement of the specimens, and the storage, the approach to the storage. And I'm showing our experiments here. The first question goes about the storage. We will test several approaches. First is direct extraction, second from fresh specimens, samples. Second is the freezing. Third is storage in setup buffer, and the fourth is the storage in alcohol. Then goes the question about sample weight. And I collected samples of peat or sphagnum, that part of sphagnum, and weighed them on the scale to show you the example. If we would take this volume of sphagnum, it weighs about 10 grams. Now it's slightly less because it stayed open overnight and it is dried already. From this test, we suggest that we would collect about 5 grams of fresh weight of sphagnum, which is this amount, and we counted the number of shoots so that our samples in the field would be near the same weight. Then goes the question about single specimen sample versus composite sample, and most kits for soil extraction work with the amount of 0.25 gram of soil, which is near this small amount of sphagnum. And this is quite small amount, but we will test whether this will work in compared to composite sample. Then we will collect the sample of 5 grams, but it will be collected not from a single location but from several localities like this. 5 shoots from here, 5 shoots from here, 50 centimeters apart, and so on. So the whole plot would consist of a series of collected individually sphagnum shoots, 50 centimeters apart or 1 meter apart, maybe, we will decide. And then they will put in one composite sample for further extraction. The next question is the extraction method. The intracellular extraction, which will include homogenizing all sample with liquid nitrogen and then collecting DNA, versus putting the sample in phosphate buffer and then extracting only extracellular DNA using the approach showed by Taberle et al. In this book. And the third question is about special arrangement, which I already covered partly, but apart from this composite sample, we will collect, we will establish 3 plots per habitat, 3 plots for hummocks and 3 plots for hollows, and in each plot we will cover slightly different vegetation community to cover microhabitat variation in this habitat. And also we will study another community of fungi from plant litter of bog plants. And the questions are near the same. I collected several types of litters and weighed them so to decide the amount, the volume or the number of leaves which we should collect in each plot. And now I counted how many leaves should be picked. And questions about storage, about special arrangement of samples were also discussed and decided. And the third part of our experiment so far is with mycorrhizal roots of Pinus sylvestris and Pinus sibirica, 2 trees grown in raised box, and I collected mycorrhizal roots of these trees and tried to calculate how many trees we would need to make to cover spatial variability and make the composite sample as well. So these questions were recommended to solve before the field campaign in this book, and we followed this good advice and tried to design our field experiment properly.